Hi, my name is Devin Enos. I'm a postdoc in Honolulu Baker's lab at the University of Washington, and today I'm going to be telling you about some of the work we've been doing towards generating the first human atlas of fetal salivary gland development. There are three major types of salivary gland that produce over 90% of the saliva in our mouth and all develop in a similar way. Development begins around 6 to 8 weeks gestation with the invagination of the oral epithelium into the underlying mesenchyme to form a bud that undergoes multiple rounds of branching morphogenesis, ultimately giving rise to a dense network of ducts and acini that will ultimately comprise the mature organ. There are multiple types of cells in the salivary gland, including multiple types of duct tissue and, namely, the saliva-producing acini. We're interested in human salivary gland development in order to better study multiple salivary gland associated diseases and developmental anomalies and to ultimately generate better therapeutic solutions which are currently lacking. And we believe that the best way to go about this would be to generate iPSC-derived salivary gland organoids as a means of both study and therapeutic development. We acquired human fetal salivary gland tissue between 12 to 19 weeks gestation, froze it, and isolated the nuclei. We then performed single-cell combinatorial indexing RNA sequencing, or SCI sequencing, and analyzed the resulting data with both SAROT and the UW-developed Monocle 3. The resulting data segregated into 21 unique clusters that were identifiable by known transcriptional markers and functional characterization of genes expressed. We were also able to perform pseudotime analysis, which computationally predicts the developmental trajectory of a given dataset through a biological process, and this revealed that the human salivary gland most likely develops as a result of transcriptional regulation of three individual developmental programs. Today, I'm going to be telling you about a branch point of one of them, the myoepithelial excretory trajectory. Cluster 4 represents basal epithelial progenitors, which give rise to excretory duct in cluster 7 and myoepithelial cells here in cluster 6. When evaluating the temporal distribution of the cells, we observed that some of the population of cluster 4 seemed to tend toward the excretory duct during the 12 to 13 week point, whereas another population seemed to tend a little more toward the myoepithelial cells in the 14 to 16 week population, suggesting that there might be multiple basal epithelial progenitor subpopulations. Therefore, we subset this cluster with more strict parameters, and we observed that this cluster split into three other clusters. And when we look at the temporal distribution of the cells within these three groups, we found that it mimicked the distribution seen in the larger plot, where the majority of the 12 to 13 week cells sit in cluster 2, 14 to 16 weeks sit in cluster 1, and 17 to 19 week is a little bit mixed, as we observe here. When we looked at top gene expression for each of these clusters, we observed that in cluster 1, this pink cluster here, we had high levels of genes associated with myoepithelial progenitor markers, while in cluster 2, this green one here, we observed high levels of basal markers, so the outer edge of a stratified duct. However, cluster 3 was a little more heterogeneous, where we found luminal epithelial markers, so markers for the inner edge of the lumen. But we also observed some basal epithelial markers, as well as, most interestingly, some mature myoepithelial markers. Since we were able to identify these very distinct markers in each of these clusters of progenitor cells, this suggested to us that basal epithelial progenitors exhibit a phenomenon known as multilineage priming, wherein progenitor cells already begin to express some of the markers of their terminal cell fates. We wanted to delve a little further into this cluster 3 here, so we decided to observe the localization of some of these markers. We noticed that the mature myoepithelial markers, like ACTA2 and myosin 3A, primarily occupied this little arm of cluster 3, while luminal markers primarily occupied the other little arm. We also observed that this whole cluster was quite enriched in genes associated with proliferating cells, suggesting that this then represents not only luminal excretory duct progenitors, but proliferative luminal excretory duct progenitors, and that these little arms could be divided into luminal duct cells, and then this early stage myoepithelial like group of cells. And we wanted to see if we could observe this population in our tissue, so we stained with the basal marker keratin 15 and ACTA 2, which marks mature myoepithelial cells. And in the earlier time points, we observed that we could see some ACTA 2 that was intercalated between the more luminal side, but it didn't seem to overlap with keratin 15 despite being a little spread out. However, interestingly, we observed that by the later time points, not only did we still see this ACTA 2 expression there, but it was brighter and more polarized than before. So we see here the basal keratin 15 expression and the luminal, the very clearly luminal at this point, myoepithelial expression.